have his homework assignment, and it mainly made it a homework assignment just to help give me a chance to expose you to a certain kind of learning strategy. And what we're going to do is we are going to draw what's called a mind map or concept map of what we want to learn about the estrogen receptor protein. And it's partly review because we've learned a chunk of it, and there's some new. The review will be the transacting domain, the ligand binding domain, and the new will be the DNA binding domain information. And so let's just get started on it. And so this we started our course with, and we said, oh yeah, here's MyoD, and we now know that MyoD functions by recruiting one of these promoter binding transcription factors, TBP, to the promoter region that will help overcome the rate limiting step for myogenic gene expression, and we'll get transcription going, and that'll lead to muscle differentiation. And we also described that this is really kind of a universal example of gene expression regulation, and we have these boxes that we could just relabel. And so this could be for our current video, this could be in a juvenile mammal, where we're going to be having the estrogen receptor bind and inhibit gene expression until estrogen shows up during adolescence or puberty and reproductive maturation in all vertebrates. And it's interesting because then we would just say that, well, instead of myogenin, this is an estrogen receptor target gene. And we have named one gene just to provide a specific example, and that's the vitellogenin or yolk protein gene. And that's a great one, right? Because it brings up, oh, well, that's not even present in mammals. We don't have yolk proteins. We don't have yolk like a chicken or a fish or a frog egg. So it just broadens our memory of, or awareness that we're working with all animals and a lot of times all eukaryotic cells in our course. So it's interesting to see that this would then be the estrogen receptor instead of MyoD. And the estrogen receptor would be back here, right? Because it would be doing stuff, and we'd have the DNA of the chromatin folded up and repressed in the juvenile, and the absence of estrogen, and then we'd have it open up here. So I could have just started the course with a different video like that. It's a little bit complicated, though, because when we zoom in on MyoD, we see it's a typical transcription factor that has two domains, a DNA binding domain and a trans-acting domain. And that's a great name, remember, because trans means genetically in trans. This is cis, the cis regulatory region. It's physically connected to the transcription unit, and this is functioning in trans. It's functioning in trans by delivering this activity to the enhancer by binding to the DNA binding domain, and that brings along this sort of dog bitey activity of having a dog on a leash and you're going to get bitten. So we're going to bring that and concentrate that activity here. And in the case of the estrogen receptor, though, we have the complexity of adding that third ligand binding domain. And so that's what we want to focus on now. And we could just imagine having our whole video introduction of the course be focused on estrogen receptor. It would just be more complicated. And we could plug that same story in with any focus on any transcription factor as well. What are two cells where it's not active and it is active, like FOXO or MYC or so forth and so on? And then what are the target genes? And then what's the rate limiting step for its activity, right? And so what I'm going to do here is we're going to draw a mind map or a concept map. And what that goes like is this, is that I remember in high school I was taught, oh, we're going to make an outline. We're going to teach you how to write an outline. And so it's going to have, Roman numerals. It's going to have, alphanumeric indications, and it's going to be organized in this structured format like that. And you're like, you've got to make a outline and maybe even hand in an outline in high school as you prepare to write, a term paper, an essay paper like that. And so it turns out that education researchers, so there are plenty of people who focus on how students learn, have found that this is not a very effective way to learn. And the reason is because this is not a very memorable thing to recall, right? Oh, it's got structure when you're actively looking at it and organizing it, but it's not a great way to learn because of it's just not a visually really memorable format like that. And you can look at your notes for all your classes, and your notes look a lot like that. And in fact, your teachers organize their notes a lot like that. And so an alternative is called a mind map or a concept map. And a mind map converts a long list of information into a memorable, highly organized diagram. And we're trying to do that a lot, and we're trying to find out. And what's the best way that your brain works? And mind maps and concept maps are slightly different, but they're almost exactly synonymous. So I'll just call this a mind map that we're going to make. And what we're going to do is make something like this in your assignment. And each assignment can look pretty unique. I'll just start drawing mine to show you, and your assignment will probably end up looking a lot like mine. So I'm going to draw mine by going like, okay, so we're talking about the estrogen receptor, or ER, at the center. And then I want to talk about the three different domains. And so I make a thing like this, and there are three of them. I'll just stick with this, and I'll just talk about the first one. And so this is the trans-acting domain. And this is a review because we already talked about it. And I think it makes sense to organize this to have two of these coming out, and like that. And some people like this, some people don't. But on the lines, I like to draw this, because why is that line there? And I like to draw no estrogen. So in the absence of estrogen, we want to move our mind out in this direction and think about everything we know about it in this direction. And then this is with estrogen. Okay, and we learned in the absence of estrogen, the trans-acting domain is going to recruit an enzyme, and so we're going to have an enzyme, and the enzyme's activity is histone deacetylase, and that is called a co-repressor because it's considered, and my typing is not, or writing is not that great. It's a co-repressor because it's a transcription factor. It's considered a transcription factor. It does not bind directly to DNA. This enzyme doesn't have the DNA binding domain. So we're bringing this enzyme in to this target gene because down here we have a DNA binding domain. So we can just add that here just because of the reality of that's how we're getting that enzyme activity tethered, right, to estrogen target gene, like, for example, yolk protein, vitellogenin gene in particular. And then down here, we have this coactivator. And the coactivator is histone acetylase, like that. Like that, okay, and it's a coactivator, and it's a transcription factor, like that. So we have that down here. And, I encourage you to make your circles big. Remember I said in class, make this big. So maybe be able to put all your writing inside the circle, like that. And you could choose to have different colors or no colors, but it's memorable to have that. And maybe different shapes, maybe boxes out here and circles out here and triangles out here. And so you're trying to create something that's memorable, that moves your brain around, but yet these are all connected, right? We want to have them connected to the estrogen receptor because they're all literally part of the protein. And so then I would suggest that then there would be another box leading out, I guess we're still here, leading out from here, that would include a term like euchromatin or open chromatin, or transcriptionally active chromatin, right? And, in designing this, I like to think, maybe I should have put this one going in that up direction, right? Where this is kind of moving me up into the upper area where I'm like, oh, it's activating. And then this other one would be moving me down toward its repressing. Because out in this direction, we'd also want to say, that the co-repressor is going to lead to heterochromatin, closed chromatin, and transcriptionally repressed, like that. So I won't write that all in there. And that's a really solid framework, and there might be some more details. And then the idea is you come back to this as you learn more. And we're not going to really learn more about these, but you could learn more about the enzymatic activity and what the catalytic domain is like. And you just keep building out in that direction, and it takes you back to here. And it becomes a really great sort of like moving in a trail, sort of like an outline, but you see it's an expanding and flexible outline, and your mind is moving out that direction like that, okay? So let's just review over here, because we're also set with talking about the ligand binding domain. So the ligand binding domain is out in this direction, and again, this is a review, and so I'm not going to necessarily cover all the details that we learned, but I would want to have a ball out in this direction, where I would like to say that maybe up here, getting this, we would have, and you could put this on here, right, is that maybe up in this direction we have an agonist, and down here in this direction we have an antagonist, right, like that. And you can also include, a definition of those terms if you need to. But in our example, we want to remember BPA. So that chemical that's in plastics that's no good. And what it is an agonist for the ligand binding domain of the estrogen receptor. It activates it. And it would be good to have this kind of direction be up, I think, and it would have been better for me to have this, this direction up, right? Because it matches up, matches up with how your mind and your sort of even your mind body is kind of thinking about the layout. As we're moving up, we're going to activate, we're going to open things up. So it's a really beautiful way of remembering this example, right? And this mind map just works really well in biology because it's so physical. And then down in this direction, we have tamoxifen, right? And I'm not going to write all the details of that, right? But, I think you would want to have for our course a ball out in this direction and say ER positive breast cancers, like that. And you might have ER negative, you might have that triple negative, and you might include a box with description, right? You might still want to have some description, explanation, definition. And I'm going to stop with these. But I think, there's a couple more details that would be really beautiful for you to add on that we learned. So this is a review. I'm not adding new stuff to that. Now I'm going to go to this one, and we're going to add some new stuff. And I won't draw it, so I'm not going to come back to draw this. But as your assignment is as you learn this new stuff, go to your, start your assignment with a review, and then move on to the new stuff and put some of these features in. Oh. Then one more thing, and that is we're also going to do this, and I should have thought about this at the start, and but we're also going to have a line out that's going to come out to this, and will be the nuclear receptor gene family. So we should have 4 lines coming out, and you might want to reorganize this, but I think this works. Just leave space for this nuclear receptor, because that's also something new we're going to learn about in this video and move on. Now, the nuclear receptor gene family, we know about that idea because of their paralogs. They arose by gene duplication. They have very similar structure, as we'll see, and it's clear that they arose from an evolutionary common ancestor. And they're such a diverse group of receptors that a lot of people like to call this a superfamily. So it just means a kind of a big, diverse gene family with sort of surprisingly different activities. So because it includes the estrogen receptor and the testosterone receptor. And you're like, yeah, I can understand. Those screwdrivers look really similar, but we really want to emphasize how different they are. It also includes the thyroid hormone receptor, progesterone receptor, the glucocorticoid receptor, vitamin D3 receptor, retinoic acid receptor. So it's a big diversity of a family doing very different things, and it's just astonishing to see they're all in the same gene family, and they have very, very similar structures. So let's move on to adding this new stuff to these 2 areas, and you can come back to your assignment and fill them in to complete it, and then send me a picture of your work. All right. And so, just another mention is now, they have a ChatGPT tool to help build a mind map or concept map. So, there are a lot of resources because, again, education researchers have found that this is a really great way to learn, and a much better way to learn than a traditional outline like that. You could build an outline because you learn how to do that, but then, or you got it in your lecture notes, but then come and put your learning together into this structured way that that researchers have found that's really how our mind works. It really moves around into our different things. And remember that you're building a resource that you will recall. You'll recall the assignment, right? And I want to add to it, okay? So again, we're working on this, and we're going to add some new DNA binding knowledge, and then some nuclear receptor superfamily knowledge like that. And so we're going to start with this, and we talked about in class about how the estrogen receptor target genes have all been identified by chip. So we've got the DNA binding domain of the estrogen receptor, that's what we're focused on, but this is looking at the enhancer, or the estrogen response element, the enhancer for estrogen target genes like vitellogenin. And so scientists were able to find all of the target genes, and they could look at this estrogen receptor element, and they line them up. So, right, we have dozens of these examples from different genes, and we're like, so what does the DNA binding domain sequence look like that it binds to? And it's surprising to see, because it's different for different genes, and there's some similarity and difference. And this is what is called the consensus ER sequence. And what it is it's an averaging. So they're trying to average and indicate what letters, what nucleotides are in this. And so this is literally that enhancer box that we've drawn. And enhancers are, you can see, some of them are pretty short. This just means that we could have any nucleotide letter in this position, but there need to be 3. It's considered a spacer between these two, and that these are the letters that are really important for the protein's amino acid side chains to make non-covalent bonds with. Here's another consensus, and what it's doing is it's showing that some letters are bigger than others, and it looks like a pretty complicated image, but they're just trying to show a lot of that averaging data in a more representative way. And it's showing that some of the letters are especially critical. So you can see that these 2 C's are especially critical. They are found essentially in all of the different target genes, enhancers, or estrogen response elements. So these C's are probably particularly important for the amino acids to make specific contact with, to be sure that this is the correct place for the estrogen receptor to bind. And it's just fascinating to see that this is a pretty simple sequence. You know, we want to activate estrogen response genes, and they have this, and the estrogen receptor binds to them, but it looks really simple and surprising. And so then let me show you this, because these are consensus sequences for different nuclear receptor family members. So that's the estrogen receptor, that's the glucocorticoid receptor, that's the thyroid hormone receptor, that's the retinoic acid receptor, and so forth. And so just look at this, because these arrows provide something interesting to point out, and I think you can see it maybe really in this one too. We go G, G, T, C, A, T, G, A, C, C. And I can kind of point it out here. You'll notice that this, and it's called a half site, has an interesting complementarity to this one, because if we do this, if we flip this around and take the complementary sequence, so G binds with C, G binds with C, C binds with A, as C binds with G, and A binds with T, you can see that that sequence, that would be the other strand, is the same as this sequence if we just flip it around. And so that's shown here by these arrows. And what it's showing is that all of these transcription factors bind as dimers, and some bind as dimers where the dimer, and I'll just kind of make up this shape for the DNA binding domain, some bind as dimers like that, and then some bind as dimers like this. And so it's a superfamily, but and they have astonishingly similar the enhancers. Like, look at the difference. The estrogen in our body does something very different than thyroid hormone, but look at the letters in their consensus response elements or enhancer binding sites. And what's the difference? The estrogen receptor requires these 3 nucleotides, could be any letter, but 3 nucleotides to create a space where these dimer is a little bit spread out apart, and this one requires no space. So, of course, you don't need to memorize the sequences, but just to be aware that there's an averaging of all of the enhancers that function as estrogen response elements, and the averaging creates a consensus sequence, and the consensus sequence often has this half site feature because of its because a dimer of the transcription factor really typically binds, and the half site often has this sort of inverse or palindrome or complementary kind of sequence to it. So we really have 2 half sites that the monomer is binding, and when you go to a half site, you're like, wow, that's a really simple sequence. This just shows that DNA twisted up, and it shows the little part of the DNA binding domain that actually does the binding. And what you can kind of see here is that they're really just 2 short parts of the protein that really are like almost like fingers sliding into the groove of the DNA double-stranded helix. And the binding domain is shown here, and there's one of these fingers and one of these fingers, and those must be very structured to hold them in very specific and stiff shapes so that they fit like fingers chemically into the grooves, because some of these amino acid side chains are going to make hydrophilic hydrogen bonds, non-covalent bonds with the DNA side chains. And in all nuclear receptors, those fingers are held in place because there's actually zinc atoms that help coordinate and bind to the specific cysteines in the amino acids of the protein and stabilize that structure. Scientists have found that the DNA binding domain of different transcription factors, which there are hundreds and hundreds of them, often fit into some categories. And so all nuclear receptors, and there are other transcription factors that have this feature too, all nuclear receptors on Earth have a DNA binding domain that includes coordinating zinc atoms. You need a little bit of zinc in your diet, like that. And it's a mineral, right, that we need in our diet because we need to have these guys held together that way. And this binding domain has a distinctive characteristics that are found in all nuclear receptors, and it's called a zinc finger DNA binding domain. And so we're just going to learn that term and include it on our map as part of the DNA binding domain, because all nuclear receptors have a DNA binding domain that's a zinc finger that just has a characteristic binding to allow it to have that very specific interaction with just a few of the nucleotides in the enhancer or response element. Then I just want to show you a little bit more. So you see there's a groove here. The way the DNA is spiraled and twisted, there appears to be a groove here. And I just want to show you this. This is just the DNA double-stranded sequence lined up, untwisted like a ladder. And here are the base pairs, and it looks like a very simple diagram and like that. And we often think of this is a ladder, and those are the legs of the ladder, and those are the rungs. In this diagram, it's nice to see this. You can kind of see that if you flex your eyes or adjust your eyes, you kind of see that it looks kind of like instead of being straight across, the rungs of the ladder kind of curve back. It looks like they're curving up, but just, flex your eyes to think of them as curving back, curving away from you. So we're climbing a ladder where the rungs are not straight across, they are curved back. And so that gives the DNA a sidedness. We have this front side and we have the back side. And because it's curving away from you, scientists call this front side the minor groove of the DNA, and they call the back side the major groove. And so just one more detail that we want to learn about the structure, and this is shown here where now we're looking down at the legs of the ladder, and they're the rungs, the legs of the ladder, and they're the rungs, and you can see there's a kind of curve to the base pairs. And what that does is it presents a certain chemistry, and that's what's being emphasized here, a certain chemistry on the minor groove side and a different chemistry on the major groove side. Now, the major groove side doesn't look like a groove here, but when the DNA is twisted up, it does. And these zinc fingers are, in this case, for nuclear receptors, binding to the major groove. And that's the last part of the structure. And of course, we're just introducing a huge field of molecular biology, which is the details of how the DNA binding domain of different transcription factors actually interacts with the enhancer and binds in a sequence-specific way to the enhancer. And the consensus element, the average enhancer, and what is that chemistry of that interaction. So that's we're just trying to introduce that, and that's as far as we're going to go with that. And you want to include all that in your mind map and just try to spread it out and have those terms: zinc finger, major groove, and a little explanation as part of your mind map. Great. So now I just want to go out on the other side of that mind map where we're going to talk a little bit about the nuclear receptor family, and we'll be done. So out that direction, that could actually be in another way, that nuclear receptor direction on your mind map. And do I still have mine up here? Yeah. This could actually be the center, oh my goodness, of a mind map that has 48 different directions leading out from it, because the estrogen receptor is just one of 48 members of the nuclear receptor family, like that. So we're drawing a mind map, and I also want to tell you about this, and we'll come back to this, but I just want to show you this. Biologists do this a lot. So here's a traditional evolutionary tree with the sort of the x-axis, kind of that line being time. And this shows the evolution. These are gene duplications that led to the evolution of the nuclear receptor superfamily in humans. So you can see some duplications led to some what people call fairly young nuclear receptors, and some are very old, like that, okay? And biologists like to do this, where they'll draw these in different ways. And this here, the colors are just to show that the evolutionary tree has really interesting biology. In this case, they're coloring in for the different types of ligands that these bind to. And there's really interesting chemistry in the different types of ligands and how they fit into the evolutionary tree of this gene family, like that. So biologists have this mind map concept a lot, and it has this functional, use here, right? There's a functional use of the lines and the circles here, like that. So let me get back here and just show you a little bit about the nuclear receptor gene family. So all of them are coding genes, right? So we have an estrogen receptor gene, or the ER gene. And when you look at the coding region, they all have the same organization because they all evolved from a common ancestor. And so you can go down the coding region and actually in all nuclear receptors find the series of the domains. So it turns out that for all nuclear receptors, there's the primary amino acid sequence, and the nuclear receptor coding region starts by coding the transacting domain, then the DNA binding domain, and then the ligand binding domain. And so there's a common structure for the genes for all these nuclear receptors because they evolved from a common ancestor. And then it happens that in these proteins, they have these real domain-like structures, because we can actually take portions of the coding region and use the domain concept to build transgenes that have different domains, like that. And so it's really a fascinating group of proteins that have tremendous medical significance and in our health and disease, and they have these common structural features. This just draws it out here, in a more visual way, like that, showing that we first come to the transacting domain, and then the DNA binding domain, and then the ligand binding domain, like that. And just another visual of that. as we go through the length of the protein. And it's just a fascinating thing. And that fits in our course into the transcription factors that require a ligand. And our example is the estrogen receptor, and it represents a great big group of 48 really important receptors in our body. We're really focusing on the estrogen receptor, but it's really important to, mention that it just opens the door to direction in our mind map that takes us into just a huge amount of molecular biology. Okay, so we'll shorten it up, and that'll be our example like that. Okay, and let me just see if I have another figure on here, and we're good to go. All right, so thanks a lot. Draw a mind map, take a picture of it. It's got some review on the 2 domains and some new stuff about DNA binding and family. Great, thanks a lot.